For today's video, we will first go over what is known about the pathophysiology of celiac disease, and then because non-pharmacologic treatments through diet modification are the first-line recommendations, we are not going to go over any specific treatments today, but we will go over how celiac disease can impact drug absorption and pharmacokinetics. So let's start off this video by first discussing the small intestines, which, remember, is the GI organ that is specially designed to maximize absorption. So the way in which the small intestine is able to handle being that primary site of absorption in the body is through its immense amount of surface area. And we have already talked about this a little bit in previous videos. The small intestine contains an intricate wall-fold network as its first measure to increase surface area. The second measure is through the addition of villi, which are these finger-like projections found on these intestinal wall folds. And lastly, each of the villi is lined with a specialized epithelial cell layer, and these specialized epithelial cells, or enterocytes, contain microvilli projections. To further increase the surface area of the cells directly interfacing with that GI lumen. Now, these components we will revisit after we discuss the pathophysiology of celiac disease. Now, celiac disease is an intestinal inflammatory disease that's triggered by the ingestion of gluten. And gluten is a protein that is found in wheat. Now, the disease mechanism still remains somewhat incompletely understood. However, there are a number of components that are thought to play a role, which we will go over today. Now, gluten peptide fragments, and gliadin, which is a glycoprotein gluten peptide, interact with enterocytes in the small intestine to induce the release of zonulin, which is an epithelial tight junction modulator. And after zonulin binding its receptors on enterocytes, this will loosen tight junctions to allow the leakage of gluten and gluten peptides across this enterocyte layer, and these gluten peptides and molecules begin to accumulate underneath this intestinal enterocyte layer. Gluten also induces the enterocyte's release of a cytokine IL-15 or interleukin-15. And this cytokine activates and recruits intraepithelial lymphocytes to the enterocyte layer to promote the immune response and initiate the damage of enterocytes. These damaged enterocytes will release TTG, or tissue transglutaminase, which is an enzyme that will act upon the accumulating gluten and modify it. The modified gluten is then phagocytized into antigen-presenting cells, or APCs, which then present the modified gluten via their major histocompatibility complex, or MHC proteins, that are encoded by the HLA complex genes. Specifically, with regard to celiac, we're talking about HLA DQ2 or DQ8. And this modified gluten is presented to helper T cells, which recognize the complex, triggering the release of multiple cytokines and chemokines. These cytokines can directly trigger inflammation and damage of the enterocytes, or they can activate killer T cells, which will then directly attack enterocytes, or signal to mature B cells that make antibodies to either tissue transglutaminase or antibodies against gluten and gluten peptides. The release of these antibodies may potentially recognize their targets at or near the enterocytes, triggering further damage to that enterocyte layer. So all of these insults together trigger inflammation and damage of that intestinal epithelial cell layer. Moving to the next slide. So all of these insults lead to a loss of enterocyte function through potentially microvilli and cell damage, or just a complete loss of the enterocytes altogether. Moving to the next slide. Additionally, this enterocyte damage and loss significantly impacts villi structures, and the result is villus atrophy. So together, this significantly reduces the intestinal surface area, which is why a number of patients with celiac disease may present with a failure to thrive due to malabsorption. Because of this loss of the immense surface area necessary to ensure proper absorption of nutrients from the GI tract. And this also translates to the absorption of medications as well. So if we move to our last slide. So remember, regardless of whether a drug is a weak acid or a weak base, the majority of drug absorption occurs in the small intestines because of the immense surface area. However, in the setting of celiac disease, with villus atrophy, microvilli loss, and enterocyte loss, the surface area is significantly reduced, which will significantly reduce drug absorption. This reduced extent of drug absorption may cause the patient to no longer be at a therapeutic concentration and not experience any benefits of taking the medication. So let's go over an example. So we talked a little bit about bioavailability, or F, before, but we will revisit the concept of oral bioavailability again today. And remember, this is just the fraction of drug that gets absorbed. So drug that makes it in over the total amount of drug administered. And this can be either a fraction, sometimes it's shown as a percent. So let's say a drug has a bioavailability, or an F, of 0.9 or 90%. So this means that 90% of the drug is able to be absorbed to potentially produce therapeutic effects, and only 10% doesn't make it in. But in a patient with celiacs with these intestinal changes, this may lead to that same drug only being 50% absorbed now, and then 50% is lost and doesn't make it in. And that 50% may not be enough to elicit therapeutic effects. Also, in a patient with poorly controlled celiac disease, the absorption of drugs may be very unpredictable, which will make it difficult to manage other conditions. These considerations will be particularly important regarding drugs with a narrow therapeutic window. And we've talked a little bit about this before, but let's revisit this. And if we look at a concentration time graph, which I'm showing you here, and say we take the curve of the drug being administered. Now, when we say a narrow therapeutic window, this means that the therapeutic concentration is not far off from the toxic concentration. And with this level of unpredictability in drug absorption and variability in terms of celiac disease control, it will be very easy for these narrow therapeutic window drugs that are being prescribed for other comorbid conditions to accidentally fall outside of the therapeutic range and not be beneficial, or kick above that minimum toxic concentration, leading to side effects for the patient. And both would obviously not be ideal. So in summary, it will be important to understand the management of celiac disease regardless of what you're treating the patient for, because if their celiac disease is not managed well and well controlled, then it is likely that their other conditions may not be therapeutically managed as well as expected.